Hi, everyone. Welcome to DataFAIR 2023. My name is Maya Stedman. I'm a solutions architect at Elucidata, and I'm going to be your host for today. For those of you who are already familiar with Elucidata, welcome back. We're so glad to see you again. And for those of you who may be new to us, we're so glad that you're here. We're back for the fourth year in a row of hosting DataFAIR. And this year, the theme is Generative AI and Life Sciences R&D for LLMs and beyond. We've got an exciting lineup of thought leaders and domain experts who have successfully transformed data practices within their organizations for more successful use of emerging tech like generative AI. So before we get started, just a few rules of engagement. You can post any questions that you have for our speakers and panelists in the Q&A section of Zoom. That's different than the chat. We do encourage you to also engage throughout the presentation with your fellow attendees in the chat. And we'll get to any questions that you have in that Q&A section in the last 5 minutes of each session. Additionally, all of the content that you see today will be made available on our YouTube channel. So if you have to pop out or if you just want to revisit some of the great talks that we have, that's going to be available in the next day or two after we finish putting this together. This is a live panel, so it should be really exciting. And if you have any additional questions for us throughout it, you can always email us at info@elucidata.io. Okay, so while people are still rolling in here, I'm going to go ahead and introduce our first speaker. So without any further ado, our first speaker is our CEO and co-founder of Elucidata, Dr. Abhishek Shah. Before starting Elucidata, Abhishek was a senior scientist at Agios Pharmaceuticals, where we had the pleasure of working together on metabolic flux, amongst other things. He has a postdoc from MIT and holds a PhD in computational biology from the University of Chicago. In his talk today, Abhishek will discuss the importance of data quality in training generative AI models and its role in enhancing the robustness and reliability of target prediction in the pharmaceutical industry. Gen AI has the potential to transform the drug discovery field. However, these models need to be trained with quality data sets before they can be productionized. In contrast to companies like Grammarly, which has access to billions of data points, the biomedical field often deals with very limited and unstructured data. Using inadequately trained models in this context can result in inaccurate predictions, unviable outcomes, and significant project expenses. So with that, AJ, I'd love to invite you to take the mic here and kick us off.
Thanks, Maya. Please let me know if I can share if you can see my screen.
Yep, you're all set.
Awesome. Thank you, Maya. Thank you, Maya. And thanks. I wanted to thank my team. There's been a ton of work that has gone in the background, and you guys will see that in the rest of the day. I also wanted to thank our speakers and panelists. And finally, I want to thank all the folks in the audience who are taking time out of their busy days to come and join us. We have had 2 events leading to this virtual event. They were both in person, thanks to the post-pandemic world. One in SF, one in Boston, and the kind of participation that we got around generative AI was yet another evidence that there's a lot of interest in the community about what this means. Is this hype? Is this a real value add? And that's pretty much the conversation that we want to lead and drive in this community. And hopefully today you will get something useful out of this. It's a very rapidly changing space. Almost every week there's an exciting paper coming out. Companies are announcing exciting things. So hopefully you will get something useful out of this, and you will see how this fits into your R&D agenda or not. Now, just to set the context, right? Biological data is growing. We all know that. We all appreciate that. We have all been, perpetrators of that, and we have all been, suffering because of that. So I'll not belabor this too much, but it always, I always find this amazing that just to compare it to something like Twitter, where the expectation is that by just in 2 years, the genomic data alone, which is one of the many types of data that scientists use to drive a discovery team in biotech and pharma companies, genomic data alone will be twice the size of Twitter, or all the data that comes out of Twitter. There are many ways to slice and dice this, but the point remains, and we all believe that biological data will continue to grow in times to come. Now, what has been our specific focus in that particular context background is that the data is available, in public domain, in your own companies and labs, but it's difficult to use. And if you see this funnel, right, there's a universe of data which is pretty broad. We can't even characterize it as to how many repositories, are out there, how many data sets are getting generated. It's very hard, right? But we all know that it's huge. Many different types of data. But if you look at the end of the funnel, there's only a small or very, much smaller data corpus that you currently focus on. And that alone is hard, right? That requires a lot of tools and expertise. But with this, new inflection point that we believe has been reached by tools like generative AI, and it is a tool at the end of the day, it's a pretty compelling and powerful tool. We believe that that funnel will shift upwards, right? So that blue ring that you see at the end, qualitatively, we anticipate in future, right? It's happening right now, but you will see more of this in the next couple of years, where people will want to use more and more data, structured, semi-structured, different types of data to leverage them, to drive their discovery programs. And that's what we would like to dig deeper into in this presentation, and also others that you'll hear about from my colleagues, and also key opinion leaders from outside Elucidata. And again, this is to stress that there's value in more data. And we have borrowed a plot from a paper that it's a preprint that came out just a few weeks back. And you see on the y-axis that the performance of the model, this was based on single cell data, human data. And on the y-axis, you have number of cells, right? So the more cells that you have, your model becomes more accurate. Again, this plot in different shapes and forms have been published by many labs. This was a recent one, perhaps a more relevant one. So we are trying to make the case that more and more data sets will be used to perform tasks for discovery teams. But these are early days, and I think there's a broad range of opinion about what this is, right? And I just saw that there was a poll that Aditi posted about where do you stand in this spectrum. And again, as I said, it's a great tool, a very promising tool. We believe that this has reached an inflection point that can create real business value, right? And then it's a very rapidly evolving space too. So we are learning about it. We are trying to understand the limitations. We are trying to understand the promise. At this point, we have shown enough evidence that, and others have too, that there's enough business value to be created out of tools like generative AI. And that's what we would like to dig deeper as to how to realize that value. What are the risks to realize that value, and how can you potentially de-risk that? That's a conversation that we want to drive, we want to lead in this community. So what we anticipate, just to paint you a picture of what we think will be more commonplace. In many ways, it has already started, right? In a couple of years, I'm talking about 2 to 3 year time frame, right? Where we see there will be a very a repository of sorts of foundational models that will come out, primarily driven from academia, which are focused on very specific problem statements. For example, gene signature prediction, cell type annotation. We have already been familiar with the protein structure prediction. AlphaFold is a one good example. Target ranking, and you can keep expanding that list of specific business question, if you will. Protein binding prediction, antibody, like you can just expand this list into a pretty broad one, right? And what will feed into these foundational models are multimodal data. Omics data, that's a good candidate to feed into these models. Assay data, data from abstracts from papers, clinical notes. So again, a whole range of like hundreds of different types of data, multimodal, would feed into these foundational models. And not all of them have to go into all the models, right? Like, so that will be a context dependent decision that groups will have to make. And further downstream, what we anticipate changing a lot as to how you consume the data. And right now we have, all of us have learned how to use tools like Spotfire to do some visualization and analysis, learn something about the data. We have also learned how to use more sophisticated tools like SageMaker, where you can run your ML model to classify patients, and so on and so forth. There's a broad spectrum there. What will change in the immediate future is we will see more examples of natural language-based, instructions-based consumption journeys. For example, you'll just want to type in plain English, like, give me data sets with this disease indication, or give me data sets which have these genes going up compared to some other gene, right? So just plain natural language instruction, which will be translated into SQL query or a code to run on your data, arguably multimodal data, right? And that we'll see more of that. Again, I think that has already started. There's some very exciting papers that are coming out on a weekly basis. Translation of language model, language instructions to code or queries has seen a lot of improvement. We have been using it. A lot of other groups have been using it as well. But we believe that this will be a very real mainstream thing that most of the discovery teams will be using in a couple of years from now. And that journey has started already. And to get there, so that all the labs, all the discovery teams can take advantage of this exciting tool that is available to them now. We have learned that there are three key things, among others, right, to focus on. One is data quality, and if you are not feeding high quality data to your foundational models, you're compromised. We'll show you some examples of that. I think building the context is very, very important. Something very simple, like you don't feed single cell data to AlphaFold, right? That's kind of easy example of like what context would mean. But I think there's more nuance, right? If you're building a protein structure foundation model, you'd also want to filter out structures which have poor R square, or you have many very contextual, very specific metrics to qualify a data. So that context is very, very important for the success of adoption of this technology. And finally, we believe any discovery team would want to be in control of the data. So they would want to know what happened to the data at what stage. And there are other aspects too, like, for example, governance, observability. In another keynote talk that you will hear today, John will talk about that. We will talk about some other aspects of it too. But I wanted to focus mostly on the data quality piece. That is a very hard problem that Elucidator has committed itself to solve. I think we have a lot of pieces, and we have the best team to really move the dial on this. And we are seeing this in the market on a daily basis as to how hard it is and how we are getting better every single day. So let's dig a little deeper. First, I'll start with some published examples of how that has been the case. AlphaFold is a very popular, well-recognized model, and this plot shows that your model improves as you include higher quality data. And the other, which is perhaps less well known, a preprint that came out just a few weeks back on single cell data, also illustrates the same point. Again, both of them are in public domain, so I'll not dig deeper into this. Now, there are a couple of case studies that we will take a deeper look into. One is around single cell, the other is around bulk RNA-seq. But the point remains, right? Like, if you were to take advantage of tools like LLM and generative AI, you would rely on high quality data. So this is an experiment that we ran. We took a corpus of around 200 single cell data sets. Again, the point was just to make the case. They corresponded to around little less than 1 million cells. That was our training corpus. We extracted marker genes for cell types of interest, and we labeled and harmonized the data sets through our poly harmonization engine. And on the plot that you see on the right, this is quite telling, right? I'll just walk you through this in a couple of seconds. On the x-axis, what we did was we deliberately added noise. And in this case, we deliberately mislabeled some clusters, right? And there's a percentage of how much noise was added. And on the y-axis, you have annotation performed and compared to human experts to find how your model perform as you add more noise. And what is quite telling is that as you have a bigger list of markers, your model performs better and better if you have high quality data. So as you move up on the y-axis, you move on the x-axis where there's less noise, higher quality data. You move up on the y-axis as well to reach this ideal point, right? And this is a very small illustrative example, and it also points out the risks of not having high quality data, right? What are the costs, right? Do you find that you have misannotated a particular cluster further downstream? And that, definitely has a huge implication too, right? So we want to belabor the point. We need to stress this more and more, that high quality data would enable you to leverage data corpuses like never before. Another case study that I want to share with you all is, we took a corpus of data sets from Geo. Geo is, a repository that requires NGS data submission for any paper that comes out. And we took this list of 1,500 data sets. They correspond to another published paper, CREEDS, which comes from Avi Mayan's lab. He's another keynote speaker. You'll hear from him later in the day as well. So we took these 1,500 data sets. They corresponded to roughly 30,000 samples, many diseases, drugs, genes, signatures. And we took the same data sets, identical data sets, and we pushed them through the harmonization engine that we have, right? So you have data at source, and the same data sets harmonized in Poly. And then we performed a bunch of text-based queries. There are different categories, 3 different categories, and I'll talk about them. And we compared the performance, right? So this was the experiment that we did to show that the value that high quality data can bring to your simple tasks, right? So let's get into this. First category of tasks that we define is the kind of query that you would make based on the metadata at the data set level, right? For example, find me all the data sets related to AML, in this case, a disease, right? Or you can say, find all the data sets that have this particular cell line, right? Or this particular drug. So any metadata corresponding to data set, we had 500 such tasks, and we performed all of them. And here I'm showing you a screenshot of one such task of this category, right? And as you can see in the screenshot, and we'll share some numbers later as well, you get some sporadic, output, right? 2 data sets showed up, they both are AML, so great. But you have no idea if you missed, other 1498 data sets that are in the corpus, right? And we perform the same task on harmonized data sets, and you get a much broader list, right? And that's very assuring. And we repeated this for 500 such tasks, right? Which require, querying based on metadata at the data set level. And we'll share the statistics of that. Clearly what you can tell from this is that, harmonized data sets outperform data at source. Now we'll move to the second type of query, which is based on the metadata at the sample level, right? And this prompt, this question in natural language, captures that, right? Find the data sets which have these drugs, in the treatment column. Again, the harmonized data set, outperforms this. And we repeated this same task for 200 such prompts, right? Where all of the tasks, all of the 200 queries were corresponding to the second category, which was, querying based on the metadata at the sample level. And finally, this was the third category, which I think is the hardest one, but a very common one nonetheless, where we queried based on the data matrix, in this case, gene expression value, right? So if we are asking, like, CGA was greater than YAP1. And if the data is not harmonized, you just don't get any output, because running a query like that is just not possible. But if your data is harmonized, as in this case, you can get a very actionable plot like this, right? Which are the data sets where this gene is indeed greater than this another gene that you have, right? and you can get the tables and all of that, right? So this is a very, very powerful tool. And if we summarize the statistics, what we see is that, the value of cleaner data gets even more valuable as you run complex tasks, right? So to summarize, the first category of tasks where we were querying just on the dataset level, we saw some impressive performance. But this performance is even more impressive if you compare the second category of task where we are querying on sample level metadata, right? And just to be clear, our source of truth is where the same data sets were looked at, where the identical queries, 500 in this case, 200 in this case, were run by 3 independent experts manually, right? Where there was no model available to them. And that is the source truth, ground truth, right? And what we see is that the automated polyharmonized engine gives you performance which are very, very comparable, as close to this red line as possible. Now, what is even more telling, which is not in the graph on this plot yet, is that the third category, where we were querying on the data matrix itself, we don't have this yellow bar at all, because it was just not possible to run such queries. And we'll compile multiple such tasks and show you how we perform compared to the ground truth. But the story that is emerging is that the harmonization adds more value as you run more complex tasks on your data corpus, right? And what I want to leave you with is, we'll release all of this in a white paper that will come out in a few weeks. But again, the idea is to show that tool like Generative AI is very valuable. It is creating real business value. For example, the prompts, the tasks that I was talking about in my previous few slides, they are real questions that you run into almost on a daily basis, right? So we're very excited to lead and drive that conversation. This white paper will, I hope, be well received and will generate many more such conversations. I'll stop here. I want to thank Maya, and I'll be happy to respond to at least a couple of questions.
Yeah, AJ, I think we have time for at least one question, and you should be able to also answer by responding by typing into the Q&A. But I'll ask you one right now.
Yes.
We get one that came in that said, do you think an LLM trained with biomedical data will be able to give similar performance with unharmonized data?
With what? With unharmonized data?
Yeah, with unharmonized data.
Yeah. So, there's definitely, if you go back to the slide, right? So it definitely depends on what kind of task you're asking, right? If you compare just these two, the performance is, much more starker for this task, which is querying on sample level metadata. So, there can be some cases where you would get a lot of value from even unstructured data, right? Or semi-structured data, but that depends on the task. What we are showing here is the more complex the task and arguably more valuable the task, you would critically rely on harmonized data.
Mm-hmm. Yeah, awesome. So unfortunately, we do have one more question in the chat, but AJ, I'll ask you to just type it out in the Q&A chat for us. AJ, thank you so much for kicking us off with this great presentation. And we'll I'm sure be hearing more from you soon. Okay.
Much.